Hello students, in the last class we studied one type of the complex tissue, that is xylem. So now we are going to deal with the second category, that is phloem. The term phloem, it was given by the scientist Nageli. The phloem, it is also known as leptome. It is also known as leptome. The term leptome, it was given by Haviland. Along with that, the phloem is also known as bast. It is very well known to all of us that phloem is involved in the conduction of food. So first of all, we will see what are the different components which are present in the phloem of angiospermic plant. 4 components. They are the part of the angiospermic phloem. So let's write the name. Sieve tube, companion cells, phloem, parenchyma, and phloem fibers. Yes. So these are the 4 components. So we are going to compare the 4 components with respect to the basic features. So let's say first thing we will see whether the cells they are living or dead. Sieve tube, living cells, living companion cells. Phloem parenchyma, living. Fibers, they are going to represent dead cells at maturity. Now the next one, whether the cell wall is thick or it is thin. Sieve tube, made up of thin cell wall. Thin cell wall which is going to be made up of cellulose and pectin. Companion cells, thin cell wall. Parenchyma, thin. Fibers, thick cell wall. And we know the thickening which is present, that is of lignin, right? Now the important functions. Sieve tube, they are involved in the food transport. For the food transport, the sieve tubes, they are helped by the companion cells. They help the sieve tube. They control the activities of the sieve tube. Phloem parenchyma, involved in the storage of several type of compounds. Fibers, going to provide strength, mechanical support. Mechanical support. Now these are the different components of the phloem present in the angiosperms. So let's see out of these which components they are also seen in the group of plant, that is the gymnosperms. In the gymnosperms, the sieve tubes they are absent. Instead of the sieve tube, there is presence of sieve cells. Right? True companion cells they are also absent. Instead of the companion cells, there is presence of albuminous cells. Phloem parenchyma, phloem fibers, they are present. So now I am going to deal with all these components in detail. So first of all, I will be dealing with sieve tube. Sieve tube. Now the sieve tube, it consists of several thin wall cells. Yes, it is formed by the joining of several thin wall cells which are known as sieve tube members. Formed by the joining of several thin wall cells which are known as sieve tube elements or sieve tube members. So just like the vessel, the sieve tube is also formed by the fusion of several cells. So let's say this is a sieve tube formed by the union of several cells, right, which are known as sieve tube elements. So these are sieve tube elements. So again, what this is going to represent? This is going to represent cross wall, end wall. The cross wall is going to contain pores. Yes, the cross wall is porous, like this. The cross wall is going to have the pores. The cross wall which contains the pores, it is known as sieve plate. Yes, this is known as sieve plate. And what is the function of these sieve pores? It will connect the cytoplasm of one cell with the other cell. It helps in the connection of the cytoplasm, right? Now, in the case of the mature sieve tube, there is presence of peripheral cytoplasm, there is presence of large vacuole, but the nucleus, it will undergo degeneration. Peripheral cytoplasm periphery की तरफ चला गया है, center के अंदर vacuole है, but the nucleus is absent. So the nucleus is absent. That's why the activities of the sieve tubes, they will be controlled by the cells which are known as companion cells, right? Now, in the sieve tube, there is presence of P proteins. P proteins, they are phloem proteins. Phloem proteins. These proteins, they are involved in the two functions. They are going to play. One, they are going to be involved in transport of material, translocation, movement of materials around the sieve tube, in the sieve tube. Second role, they are also involved in healing. What do you mean by that? अगर sieve tube के अंदर कहीं पर कोई क्रैक आएगा, कोई ब्रेकेज आएगी, that part is going to be sealed with the help of P protein. So the P proteins, they are involved in sealing of cracks. So you can say they have role in healing. They are involved in sealing of cracks or sealing of the areas of the sieve tube which have shown injury or breakage, injured areas, right? Along with that, I would like to add one point over here. These proteins, they are involved in the transport, translocation, and this translocation, it is dependent upon ATP. These are ATP dependent proteins. Now, during the unfavorable season, you know, the metabolic activity in the plant is very low. If the metabolic activity is in the plant low, so the food requirement by the plant is less. Food requirement by the plant is less. So it means under such situation, the transport of the food through the sieve tube, it should decrease. So to decrease the transport during unfavorable condition, these pores, they are plugged. Yes. To decrease the transport during unfavorable condition, these pores, they are plugged. Pores, they are blocked with the help of a chemical which is a carbohydrate, that is callose. They are plugged with the help of callose. That's why they are also known as callose plugs. Got it? They are the callose plug. Formed during which season? Unfavorable season, late growing season. But during the favorable season, spring season, on the plant there is formation of new leaves. For the formation of new leaves, there is requirement of food. There is requirement of resources. So under that time, these callose plugs, they will be dissolved so that the food requirement, it is fulfilled. Are you getting this? Callose plug बनेगा कब? Late growing season के अंदर. Right? तब ये बनेगा. Yes. And during the spring season, जब प्लांट के ऊपर नई पत्तियां बन रही हैं और उन पत्तियों को बनाने के लिए बहुत सारे रिसोर्सेस की जरूरत है, फूड की जरूरत है, then these plugs, they will be dissolved. They will be dissolved so that food requirement is fulfilled. So you got this? Now after the sieve tube, now the next component, that is the companion cells. So now we are going to deal with the companion cells. On the site of sieve tube, to control the activities, to control its activity, there is presence of companion cells. They are thin parenchymatous cells. Parenchymatous cells. So let's say this is our sieve tube. This is our sieve tube. Yes, this cross wall is porous. Come on, tell me what this porous cross wall is known as? Sieve plate. Yes, now around on the sides there is presence of companion cells. So these are companion cells. Now the cytoplasm of the companion cells and the cytoplasm of the sieve tube, they are connected. Yes, you can see here साइटोप्लाज्म कंपेनियन सेल का और साइटोप्लाज्म जो सीव ट्यूब है इसका कनेक्टेड है with the help of this pore. This pore, they are known as pit fields. These are the pit fields which are present in the common longitudinal wall of sieve tube and companion cell. Yes, these pit fields, they are present in the longitudinal wall. Now, as we have discussed in the sieve tube, the nucleus is absent. So, the activities of the sieve tube, they are controlled by the nucleus of companion cell. It controls activities of sieve tube. I would like to add on one point here. The companion cells, they maintain pressure gradient inside sieve tube. A pressure gradient they maintain. Let's see how this is maintained. From the MSC that is mesophyll cell, food in the form of the sugar is transferred to companion cell. Sugars, it is transferred to the companion cell. From the companion cell, the food is transported to the sieve tube. जैसे फूड सीव ट्यूब के अंदर आएगा, सीव ट्यूब के अंदर ऑस्मोटिक प्रेशर इंक्रीज हो जाएगा. फूड enters the sieve tube, osmotic pressure is going to increase. Osmotic pressure increase होगा. So you can say water potential will decrease, right? Water potential कम हो जाएगा, water content कम हो जाएगा. So what will happen? The sieve tube will take the water from the adjoining area where the water content is more. It will take water from adjoining area where water content is more. What is that? That is the xylem. तो इसका मतलब जाइलम से जो पानी है वो सीव ट्यूब के अंदर भरना शुरू हो जाएगा. जाइलम से जो पानी है वो सीव ट्यूब के अंदर भरना शुरू हो जाएगा. सीव ट्यूब will absorb water. वाटर एब्सॉर्ब करने की वजह से बनेगा टर्गर pressure. This formation of a pressure. इस साइड ऑफ सीप ट्यूब के टर्गर प्रेशर ज्यादा है, यहां पर टर्गर प्रेशर ज्यादा है. At the other end of the sieve tube, the turgor pressure is less. So here the pressure is more, here the pressure is less. There is formation of pressure gradient. Yes, there is formation of pressure gradient. And due to this pressure gradient, now the food is pushed from one end of the sieve tube to the other end. Are you getting this? यहाँ पर टर्गर प्रेशर ज्यादा, यहाँ पर प्रेशर कम, gradient बना. Due to this gradient, now the food is pushed from this end of the sieve tube to this end. So this is going to help in formation of pressure gradient, which results in movement of food. So this is very much clear to all of you how the companion cells, they will maintain pressure gradient. Now these are the two components which are present in the angiosperm. Let's immediately compare them with the components present in the gymnosperm. In the gymnosperms, there is presence of sieve cells. These are sieve cells. So you can see elongated with the tapering ends, going to have thin wall. So no sieve tube, elongated cells with tapering ends, thin cell wall. Right? Now for the transport of the food, again the cell wall should be porous. Right? Now here there is no cross wall. No cross wall is there. So now the pores, they are present on lateral walls. Lateral walls. They are going to have pores. Like this. lateral walls, they will have the pores rather than the cross wall. So now these pores, yes, यहाँ पर जो ये pores present है, they are not going to be known as sieve plate. Rather, they are known as sieve areas. Got it? They are known as sieve areas. Again, in the sieve cells, at the mature stage, the nucleus is absent. Nucleus is absent. So to help these cells, to control the activities of these cells, there is presence of adjoining cells. कुछ help cells present है, पर ये जो help cells present है, they are known as albuminous cells. Got it? They will control the activities of sieve cell. So let's immediately compare between angiosperm and gymnosperm with respect to these two components. Angiosperm, sieve tube made up of several cells, right? Gymnosperm, sieve cells, they are only single. They are single cells. In the sieve tube, the pores, they are present on cross wall. Here, the pores, they are present on lateral walls. Side की walls के ऊपर pores present है. यहां पर जो पोरस क्रॉस वॉल है उसको आपने क्या नाम देना है? सीव प्लेट. और यहां पर आपने उसको नाम देना है सीव एरियाज. राइट? यहां पर न्यूक्लियस जब सीव ट्यूब में एब्सेंट है, सो हेल्प इज परफॉर्म्ड बाय कंपेनियन सेल्स. And here, the help is performed by albuminous cells. Now, the albuminous cells, they are also performing the function of companion cells, right? They are helping the sieve cells. In the same way, the companion cells, they are happy in the sieve tube. But still, the albuminous cells, they are not known as true companion cells. Why it is so? Yes, so now we are going to answer this question. Why the albuminous cells, they are not known as true companion cells? Now the companion cells and the sieve tube, they derived from same mother cell. They derived from same mother cell. So they are known as sister cells. So they are derived from the same mother cell. So they are going to be known as sister cell. So they are ontogenetically related. You can say they are ontogenetically related, right? But about the sieve cell and albuminous cells, now they are not derived from same mother cell. They are not derived from same mother cell, so they are non-sister cells. They are non-sister cell. That's why the albuminous cells, they are not known as true companion cells. Got it? Ontogenetically, they are not related. That's why they are not known as true companion cells. Got this? Now, after this, now we are going to deal with rest of the components of the phloem, that is the phloem parenchyma and phloem fibers. Phloem parenchyma made up of elongated cylindrical cells the cytoplasm of the cells, they are connected, is connected with the help of plasmodesmata. Cytoplasm is connected with the help of the plasmodesmata, that is a pore between the two plant cells. Now these cells, they are involved in the storage. Function is storage of food like the one we have studied in the xylem parenchyma. Along with that, involved in the storage of resins, mucilage and latex. A very important thing which need to be learnt here, that this phloem parenchyma, in most of the monocots, it is absent. Very, very important. In most of the monocots, the phloem parenchyma is absent, as well as it is absent in few herbaceous dicots. So till now we can say that in the monocots we have studied, lateral meristem is absent, cambium is absent, collenchyma is absent, and the phloem parenchyma is also absent. Now the phloem fibers, which are also known as bast fibers. Phloem fibers, also going to be known as bast fibers. Fibers, they are sclerenchymatous fibers, thick wall made up of lignin. Now some of the plants, the phloem fibers, they are commercially important. Commercially important. So I will be writing the name of the few plants where they are of commercial value. One is flax. Flax. The scientific name for the flax is Linum. It is a Linum species. Now, second one, that is jute. Scientific name is Quercus. The next one is hemp. And the scientific name is cannabis. Yes? So it's really very important to learn all these three examples where the fibers, they are commercially important. They have commercial value. Along with that, this point that in the monocots, phloem parenchyma, lateral meristem, and what else? Collenchyma is absent. After looking into the structure of the different components of the phloem, now I will be dealing with the types of phloem on the basis of origin. So now I will be dealing types of phloem on the basis of origin. Primary phloem, and secondary phloem. Now the primary phloem is that phloem which is formed by procambium when there is formation of primary parts of the plant. When the primary parts of the plant, they are forming, at that time the phloem which is formed by the procambium, this is known as primary phloem. Now the primary phloem, just like the primary xylem, it is divided into the two types. It is of two types. जो primary phloem सबसे पहले बनेगी, that is protophlloem, which is formed later, that is metaphloem. Protophlloem, that is first formed. Primary phloem. Metaphloem, it is formed laterally. Primary phloem is formed before the plant organs, they have shown the growth. Before the plant organs, they have completed the growth. But the metaphloem is formed when they have completed the growth. Now the protophlloem, it is formed when plant organs, or I should write before plant organs have completed their growth. Completed growth. Right? The metaphloem is formed when the plant organs have completed their growth. Formed when plant organs have completed their growth. Right? Now when the protophlloem is formed, the plant is very small. Yes, the plant is growing. It is small in size. Under such condition, the elements, whatever the sieve elements which are present, they are smaller in size, as well as the lumen is narrow. Going to possess smaller size elements. And they are going to have narrow lumen. But when the metaphloem is plant, now the plant is tall enough, so the elements which are formed, they are longer. They are longer, and now they are going to have broader lumen. Now in the case of the protophlloem, let's see what are the different components of the phloem which are present. There is presence of sieve tube. There is presence of phloem parenchyma. But the fibers and companion cells, they are absent. Fibers and companion cells, they are absent, as we have seen in the case of the protoxylem. प्रोटोजाइलम प्रोटोफ्लोएम जब बनेगी, प्लांट की हाइट बहुत कम होगी, so the mechanical support which is required, that is lesser. That's why there is no requirement of the fibers. Along with that, during the young stages of the sieve tube, the nucleus is present. If the nucleus is present, so it means now there is no need of companion cell to control the activities of the sieve tube, as the sieve tube already contains the nucleus. Now coming to the metaphloem. Now all the four components, sieve tube, parenchyma, fibers, companion cells, they are present. Now in the protophlloem, the sieve areas, or you can say the sieve plates, these less developed. Sieve pores less developed. Here the sieve pores more developed. Now the protophlloem, it is short-lived. Yes, it is short-lived. It is crushed by the surrounding tissue due to the primary growth. जब आसपास टिश्यू फॉर्म होगा प्राइमरी ग्रोथ के टाइम पर, दिस प्रोटोफ्लोएम इट विल बी क्रश्ड. मेटाफ्लोएम इट इज लॉन्ग लिव्ड, but it can also be crushed, but its crushing will occur during secondary growth. Are you getting this? This protoploem, it can be crushed during primary growth. Primary growth के time पर the metaploem, it will be crushed, but the crushing will occur laterly during the secondary growth. Got this? Now, after the primary phloem, now we are going to deal with the secondary phloem. Now, you can easily answer this question. How this phloem will be formed? Just like the secondary xylem, this phloem will be formed by vascular cambium. Formed by vascular cambium during secondary growth. Just like the secondary xylem, it is again divided into the two systems. Yes, do you remember that? The system which we have studied in the previous class. All the components of the phloem which are present in the plant vertically, it forms axial system or vertical system. Right? All the components of the phloem which are present in the plant now horizontally, they are going to form radial or horizontal system. Got this? So now we have to see the components which can be the part of the axial and radial. Yes, tell me. Sieve tube, how they will be placed? This or this manner? Yes, sieve tube. They are involved in the transport of the food from the leaves to the root and the other parts of the plant. So they are going to be placed vertically. Sieve tube के साथ साथ companion cells. Then phloem fibers and type of phloem parenchyma which is present vertically. It is known as axial parenchyma. Right? And the type of parenchyma which is present in the plant horizontally, radially, this is ray parenchyma. Ray and axial parenchyma both involved in the storage of food. So after looking into the complex tissue, that is the phloem, now I will be dealing with the tissue system.